Right, so, how we validate antibodies, we've been doing it the same way for many years. So we've got this, that's why the portfolio is the size it is, because we've been doing it the same way. We start off, obviously, by immunizing rabbits. They're all rabbit monoclonals. They go through that process, right? So, an important part of that is getting IHC into FFPE, so we're going into tissue microarray as soon as we can. So, you come out of that immunization process, you have thousands of possible clones. You screen those down until you have a reasonable, often 100 or so clones that you're then thinking might be specific or sensitive for that marker, right? And you then got to screen them. So we then take a tissue microarray approach, FFPE, multiple, tumor or normal, often mouse and rat. It kind of depends on what marker we're really looking for, but
exactly. Where's it going to be clear positive, and where's it going to be a clear negative?
Exactly, and we know what it's going to be. We know whether it's going to be a nuclear marker or a cell surface or whatever it might be. So we kind of go into that first, and so we can then pull out what we think is probably the most likely clone. Sometimes we'll be lucky, we'll get, that's the one. It's got the most, it's got the best signal to noise, best sensitivity. And then we go through the process all over again. So that's that kind of screening process down from 100 down to that perfect one.
Right.
And bring that into a full validation stream. And that full validation stream then is, if I remember right, 17 different cancer tissues.
I've noticed that over the time that your extra validation that you're doing on the back end has increased a lot. Like we're seeing a lot more knockout work, we're seeing a lot more multiple tissue work, we're starting to see, we see secondary only negative controls, we see more negative control tissues. It's really been great to see it all kind of amplify.
Yeah, and so it's kind of dependent on the target and where we have got a knockout, we'll absolutely put it in.
It's great.
And that gives us that kind of added value and confidence, really, that is definitely being in that specific area.
Yes.
But I mean, even if we're going down a tissue microarray route, again, we know that it's going to be prostate cancer, it's going to be breast cancer, whatever it might be. So, we then do that either in a tissue microarray, always FFPE, always high quality tissue, working through into normal tissue, etc. So that we can then go through a whole titration stream.
Right.
So that we can work out, where the best signal to noise lands. And we do that on an automated platform.
Right.
And work our way through. And so then it always then comes out with the same validation stream.
Right.
So if we can get a good positive negative control in there, being it whether that's cell specific or we can do overexpressors, etc. So we will try and do that, probably usually in the kind of earlier phase. So we'll do an alkaline phosphatase test, so we'll on section. So we'll do alk phos, we'll knock it out. So it's phosphor specific in the sense that we know it's definitely a phosphor.
Exactly.
Right. So it's not, we're not going to the level of it. We know it's a phosphor 229, whatever.
Right, but you know that from the initial epitope that you used to immunize your rabbit.
That's coming from the immunization. So that's the design of the experiment right at the beginning.
Exactly.
And then we determine that it's a true phosphor mark against the total.
Yep.
And often we'll add in all the other validations on top. So we'll be doing with some blot at the same time. Again,
flow,
stripped, positive, negative, flow, ICC, IP, etc.
Which is great because it's very application specific. Like you'll tell us this antibody is very specifically good for this application or that application, not recommended for this application, which means you're getting the antibodies that you need for the application you're working on.
Exactly. Yeah. Wherever possible, we'll add in mouse and rat. So in the TMA, we have human, we have normal, and we've got mouse and rat. We always do mouse and rat. That's a key species. So we've got obviously in an IO area, there are people working in. They're working.
Oh my gosh, so many different organisms.
But they're going to bounce into mouse because that's a key development piece, and then they're going to go into human. And in an ideal world, you want the same antibody. It just makes it easier.
It's so much easier. Yeah.
So if we know that it works, or we can at least state yes, it does, or no, it doesn't. It's not just based on immunogen.
Right.
going, well, we think
But
it's
also
100%.
defining that specificity can be important because you think about a mouse transgenic model, you actually need to have a human-specific version of like APP or something like that. You need to be able to tell the difference between that and the mouse native protein. So it's really great to get that exquisite specificity of the mouse and the rat and the human so that you know you can make those modifications or pick those specific antibodies to attack those kind of questions.
Yeah. And trying, wherever possible, we're moving towards more EDTA because we know, for the cells want to have that one hit.
Yes.
You want to do one retrieval.
Right.
And you know all your markers are going to come off of that. And that really gives out the confidence.
And it gives you confidence, especially in these FFPE tissues, which are so many clinical samples are coming in as FFPE. It's critical to work in that tissue.
Yeah. And getting that retrieval right, depending on, hopefully everybody's fixing it in a really good reproducible manner.
Yep.
It will work, it'll just work out of the box. It'll work
Absolutely.
out of the box of our cells and it'll work out of the box.
Good clean tissue slices on a slide, works
yeah.
right out the box, beautiful sequencing data coming off the back end. Hallelujah.
What I really like about what you do is you add that extra layer on top. You do a lot of testing in your side to make sure that, when we're looking, if you like, unconjugated, naked antibodies, that we can take it only to a certain stage.
Right.
But you take it that extra level.
Right.
And you are confident that it works in your own
And we
assays.
replicate some of what you do, right? We're putting a DNA barcode onto the backbone of these antibodies, but then we're also going into TMAs and we're going into homogeneous cell lines where we know the proteomic profile of these cell lines from other orthogonal databases. So then we're able to kind of replicate that long-term screening. We just do it in the multiplex, right? And we're also testing, phosphospecificity with phosphatase, our slides and checking the correlation between the two. And so again, it just adds to that confidence that we have both your screening data, we have our screening data. Yours is a singleplex, ours is a multiplex, but we have all that confidence of the specificity and sensitivity that we're able to gain.
Yeah, but you've got to do them both. I mean, that's the thing. It's all about cell lines. Cell lines are brilliant.
Yeah. But you need the TMAs too.
You have, because it doesn't give you where the background levels are. So you're only really getting that one thing as opposed to all the rest of the stroma, all the rest of the material that can have a non-specific background.
And all the different organs that you might be encountering, all the different tumor types you might be encountering. The cell lines are nice because we're able to use these orthogonal databases to know exactly what should be a yes, no, right?
Yes.
Think of it kind of like those tests we used to take, a bubble in the right answer kind of thing. It's cool.
If they're right.
If they're right. But we work with high confidence proteomic databases, so I feel pretty confident there. But then you're right, we need the TMAs because that's what the customers are using, right? They're in their tissues, they're in their different tumor types, they're in their different organisms, and we have to make sure that everything works like it would work in their hands every single time, right out the box.
Yeah, absolutely.
Yeah.
Right, so I mean, we've recently implemented a method of biophysical QC, and in that biophysical QC, we do three main things, but it's about identity. So we use LCMS to try and make sure that we know it's a recombinant monoclonal, right? Yeah. We know what we're putting into those cells to kind of create the antibody. We know what the heavy chain and light chain size is, and we can measure that. We know exactly what comes out from that setting, right? And then the two other things are around purity and aggregation. So we got a couple of ways of measuring aggregation. One using dynamic light scattering, the other one actually just HPLC, which also measures purity. And we can use both of those to then see what kind of purity of population have we got, and do we have an aggregate coming through that process?
Right.
Are the antibodies binding to each other? And with the confidence that we still have all the application data at the end. So we know the IHC is still working, but we know exactly what is in the tube, we know it's exactly the right identity, it's the right antibody, and then it will come to you guys for your conjugation. Again, you've got, you know what's in there, you know you're not going to get some kind of weird conjugation result.
And from our perspective, we purify our antibodies after conjugation so that we know we don't have a bunch of free DNA floating around, and we also aren't having proteins aggregate on our hands. We use both a combination of chemical methodologies as well as filtration to make sure that we don't have any aggregates coming into our final product. And that way we know that when it goes onto the slide, you're not going to get any weird protein aggregates that are going to give you false positives. You're going to get a nice, clean distribution of the antibodies across the surface. We're working on a pan-cancer atlas, right? Not every cell is going to have all of the antibodies on them. It's going to be a subset. So really what you're getting is in each individual cell, you're getting a subset of those antibodies, not everything all at once. And so we just don't really see those kind of crowding, that people I think sometimes are fearful of. It just doesn't seem to happen. And that's probably because we are hitting so many different cancer types and immune types that, per tumor type, we get about, out of our 570 panel, we get between 250 and 400 protein targets coming up, which means the antibodies are bound. And that seems very, very doable, especially if you're not using really high concentrations of antibodies, which we don't.
And they're not all, they're not all cell surface. They're not all nuclear.
Right.
So they're all scattered
They're all over the place.
entire cell. So the likelihood of them being bounded to the same kind of
unless they're literally on neighboring epitopes.
Exactly.
Yeah.
In which case you might be doing it a different way. I mean, that's the thing. Yeah. It doesn't necessarily work that way.
No, the only time I've ever observed it is when we've done we've looked at a pan and a phospho that literally on a epitopes that overlapped with each other. And then, you can imagine why one's bumping the other off or preventing them from binding. But other than that, everything's actually just more space in the cell than I think people give it credit for.
Yeah, exactly. And they do mix well, and it's not like they're specific antibodies. So they're not going to bind to his neighbor. There's no reason why we should get that kind of binding. And we make these things called multiclonals, in which case we're taking multiple recombinants all into the same tube. We don't suddenly find that the aggregation is any different, that they bind to each other. That doesn't happen.
Yeah, you
it
have to
happen in a polyclonal. Why would it happen in a multiclonal?
Exactly.
Why would it happen in your assay, which is a really big
area? Right.
really
You actually have to
laboratory.
concentrate protein down pretty far to actually start to either get aggregation or you actually hindrance with each other.
Yeah, absolutely.
Yeah.
And in our aggregation experiments, we've done 70% of our catalogue now has gone through biophysical QC. Our failure rate is at 0.8% for aggregation. They just don't aggregate.
Yeah.